All right, so when we are saying that a gene got expressed, it means that that recipe got executed, right? Because the gene is a recipe, is some instruction on how to make something. So the processes for that are transcription and translation. Not every gene needs the translation step, but I try to tell you here, when a gene encodes a protein, then it needs translation. So some genes don't encode a protein. They're the genes that encode ribosomal RNA, transfer RNA. Those genes are RNA genes. They don't encode a protein. Then translation is not necessary for you to say that that gene got expressed. But most genes on the cell, they encode the protein, so translation is needed. So how do we predict genes using the homology approach? We want to compare the sequence we have with the database of genes. So you could do this, but if you compare your sequence you have with the nucleotide collection, a nucleotide collection has genes in it, but also has a lot of non-genes. So this is saying all these are different databases, and let's see, excludes whole genome sequencing. But okay, the point of this nucleotide collection is to say that in there would be genes and also non-gene sequence. So if you're going to predict genes, you could use this, but you're going to get a lot of hits that are not genes also. So is it wrong to use that? No, but the shortcut is that you narrow it down to, I want to compare my sequence to a collection of sequences that I know are genes. So if there's a RNA molecule, then you know that it came from a DNA sequence that got transcribed. And if that DNA sequence got transcribed, that's the definition of a gene. So you want to compare with that. The whole shotgun context, that's going to give you a lot of genomic sequence. So genomic will have DNA that's not a gene, so don't check that. This, you can think about it as analogous to mRNA. So, and let's see, this one is the protein data bank. So that would be sequences in the protein data bank that we know end up as protein. And okay, this one too, because these are protein sequences. So all the point of this exercise is to say you have your genomic DNA or some DNA sequence that you don't know if it has genes or not. How would you find genes by homology? You look, do I have any sequence in my query sequence that resembles mRNA or ESTs, which you could think about, it's mRNA also, or nucleotide sequence that is in the protein data bank, so that nucleotide sequence that got translated into protein, or protein sequence that is on this database. So that is the approach of homology.